Recording. There we go. That's the danger of handing over the controls to someone else. So, just as I said, any problems, pop them in the chat. But please put your questions as we go along. And I'm going to hand over to Sam, who's going to introduce himself, explain what he does, and show you around the laboratory. So, over to you, Sam.
Hello. So I guess I do my potted history of my CV. So I'm Dr. Sam Aoudjane. My first degree was in genetics, which my undergrad degree. My second degree, my master's, was in like drug development, and then my PhD was in biophysics. And I suppose my specialization for my PhD was building instrumentation. The main focus of my PhD was trying to build very small scale qPCR machines. Everyone's heard of PCR tests and now, right? So that's what I was doing, was building a qPCR machine that was entirely laser based. So you may be aware, depending on how much you read about this, that PCR is about thermocycling, so changing the temperature of samples. And I was doing that with infrared lasers, and then measuring like the amount of DNA amplification with other lasers. So it was all optically mediated. So my background is kind of like in genetics, but then also in optics, instrumentation design. And I'm going to take you around today where with some element of that, we're going to be talking about. This lab is mostly fluorescence based measurement of intrinsic protein fluorescence. So proteins are made out of amino acids, and some of them have this property of fluorescence, which we can measure as like a way of measuring the state of the protein. So you could use that for measuring the state of some immunoglobulin you're producing, a lentiviral vaccine, you know, these things are kind of famous at the moment. So that's my background and what I do. And I thought I'd do this because I think most of the other talks are going to be on people doing sort of more straight biochemical engineering and biology, whereas this is more about instrumentation design, as physics and physical systems engineering. And so maybe some of you will find that interesting, I don't know. But so I'm gonna swap to the other camera, because unfortunately there are slides. You thought you'd get away with no slides, but you didn't. So just to explain basically like how fluorescence works, like you have, I hope you can see this. So if you imagine this is, well, you don't have to imagine, this is what they call a Jablonski diagram. So you have the energy on the x-axis, and then you imagine, time on the x-axis, and then the energy state on the y-axis here. So imagine a system, it's a constrained system of double and single bonded carbons that can absorb a photon of some high energy level. So that means some low color in the spectrum, blue or ultraviolet in this case, right? And then some energy is lost by vibration into the movement of the molecule, and then a different color photon, a lower energy photon, a higher color, green in this example, is emitted. So what we have is that you throw light of one color into the system, and then a different color light is emitted, right? So this is the process of fluorescence, right? So in a protein, in this example, this is the structure of a kind of what you might say a test molecule, BSA, the bovine serum albumin, and I've highlighted here the tryptophan residues. The tryptophan residues have this process of fluorescence that they can undergo it. So if you chuck UV photons at tryptophan residues, they will emit sort of bluish photons, and like the state of the molecule, like how denatured it is, like how tight the structure is bound, will change like how that process works. Particularly if we what we'll be talking about later is the fluorescence lifetime, like how long it takes for this decay back to the ground state from an excited state all the way back by emission, like how long that takes is dependent on like the electrochemical structure surrounding each of these fluorescent residues, right? So in order to sort of do that, in the first instance, we're going to use this fluorescence spectrometer. So this is a device that has, if you imagine a light source that's inside the box, I'm not going to take the box off because it's kind of dangerous, but a white light source and then it has like, if you can imagine a prism that allows you to select what kind of light you want to throw in, and then I've got in here a sample. Oh, where's my camera? It's on the other side. So this is the bovine serum albumin in solution here, right? And then I'm going to put that in the spectrometer, and then there's a detector and another. If you don't, these are just Allen keys, but there's another prism which we can move to select wavelengths of light that we're measuring, and then a detector that will measure that. So we'll just fire that up quickly. So if you see, I'm going to set up this measurement now. I'm going to chuck in 260 nanometer lights. That's sort of near UV. I hope you can see that. I'm going to then measure from 300 nanometers to 450 nanometers. So that's kind of like in the like violet, purpley kind of high energy visible light wavelength range, right? That was a long way of saying that sentence. So I'm just measuring that now, taking a spectrum, and this is the intrinsic fluorescence spectrum of the tryptophan residues that reside within BSA. And this is I don't know, maybe not the most interesting thing you've ever seen, but it gets more complicated in a moment. So we take in a spectrum. So we say that this we've measured the fluorescence of this protein. Now we know that it is in fact fluorescent, right? So now we're going to move into is measuring the fluorescence lifetime, right? So get rid of that. Now I'm going to have to try and not mark this up because I have to change settings on the software on the fly here. It's going to take a minute to start up. Maybe I should go back to my face. I wonder if would it be possible? Can I do it like that? So I'm just going to set the measurement up here.
Yeah, sure, I can see that.
That is. Right. So I'm going to measure the fluorescence lifetime now. Set the bias on that. Oh, it's not working. What have I done wrong? Oh, I didn't turn on. So now instead of using the light source that was built into the thing, I'm going to use an ultra fast pulsed LED, which is going to chuck packets of ultraviolet photons in at the sample. And then this more sophisticated optics is going to measure very quickly the time that it takes for photons to be emitted. So we can measure the fluorescence lifetime of the thing. Start again. Right, so now it's actually working. So it's what this is it's measuring the time that the first, once it emits a pulse of light at the sample, it will then measure the time it takes for the first photon to arrive, and it does that for every pulse, and it's working about 1 megahertz. And you can see that there's a characteristic decay to that, right? As it's more likely that photons will be emitted quickly, and so it's an exponential function, and you can fit an exponential decay to that and get a mathematical description which is related to the electrochemical state of the protein, right? So if I was to change the protein, it would have a different decay rate if I put a different protein in there, or if I was to heat the protein up and it was to denature, then it would be you just get a mathematical description that involves an exponent, and then the number that that you would get from fitting that would be different, right? So this is the key principle that underlines. I mean, that's done now. That's just underlying like how you would measure fluorescence decay, right? So now, Kim, I really hope that the Wi-Fi works in the back here because I got to go into the laser lab now.
Okay. So just let us know how that data would be used. How that information would be applied, and what the next stage of the results of that test would be. So once you've got that information, how is it actually? Oh, okay.
So I'm just going to move on to the instrument that we built here. So if we have a chromatography system thing, I don't know if I'm pitching this too hard because are these guys undergrads or not? Like,
I think it's fun. What we'll do is if we need clarification, we'll leave it up to people to answer the questions. We'll come to questions in a bit.
Okay, so this is a chromatography system that is used to pump protein samples from this robotic thing that selects them out of vials and then injects them into a chromatography column. like you will have done, I guess, chromatography at some point in your science education where proteins or some molecule is separated out by some property of it. In this case, I don't know, size or something like this. And then that's injected into, so then this is the system here that we built. So we have an ultra fast, high energy UV pulse laser, and we send packets of UV light through these optics, right? We use this little detector to monitor like the stability of the beam, and then that's directed by these optics, collapsed down by this lens onto this sample delivery tube. So the sample's pumped from the chromatography system through, right? And then I don't know if you're able to see the inside of this thing. So then this is a reflector that's shaped like an ellipse, which focuses the fluorescent light down onto this detector here, this ultra fast detector. And so we measure the fluorescence lifetime of the protein species, you know, with this, and then that's sent to a very fast oscilloscope, and then that's recorded on the computer. And then if I go back to, see, this is where I'm going to try and answer the question, right? Go back to the, so this is results from our latest paper. So in chromatography, you would have like, you would pump samples in, and you're trying to purify something. You're trying to separate things out, separate like complete vaccine, you know, particles from incomplete ones. And normally a chromatography detector only measures the absorbance of, like, you throw light at the samples as is transiting through the detector, and it like if it blocks the light, then that's absorbance, and you only get one piece of information. But by getting the fluorescence lifetime, we can extract a different proteins are eluting coming off the chromatography system at the same time. So we're building new instrumentation that doesn't exist anywhere else, so that we can discern different proteins as they're coming off protein purification processes, which is relevant in biochemical engineering, because you're trying to produce a pure product. You want to know what things are being produced and what impurities are there. And so if you can gain more information by measuring different properties of it, in this case optical properties, you gain more information. You do that by having not just cutting edge like instrumentation that doesn't exist anywhere else, like, but that's done by a load of data processing layer. So that's kind of what I was going to show today. It was a like instrumentation that already exists, and then stuff that, you know, we built, you know. And I'll answer any questions.
That's fantastic, thank you. So if anyone's got any questions about that, please use the Q&A box to post them, and we'll go through them now. While we're waiting for some questions to come through, we don't have to, by the way. If we answer your question, if we address everything, that's brilliant. But just whilst we give people a chance to answer some questions, just a quick thing to ask Sam, can you just briefly mention if there's any way undergrads get involved in this kind of research, or just give us an overview of how you work with undergrads?
Well, I mean, so mostly, like, I'd say that mostly we have some undergrads in the lab, and we'd like some more, you know. There's certainly opportunities for doing that. Like, I think it would have to be really undergrad, like, final projects, because we would be aiming for people to, you know, like, to, we're aiming to publish results. This, like, this wouldn't really be a training, so much a training exercise. It would be, you know, starting to actually do work. Like, I think the main thing that we would be doing is about protein formulation. So protein drugs, you know, like, you can mutate them for stability, but then it's also a large part of it is like how the drug is formulated. So the buffering conditions, you can put different excipients, like the success of the mRNA vaccines. Really, one of the things that was quite stark about that was that a lot of it was about how the mRNA was kind of packaged, which is formulation. And we have lots of projects about, like, formulating protein drugs. And then you could measure, like we use these techniques of intrinsic protein fluorescence to measure the state of the protein, the internal electrochemical state, and how that's affected by the formulation. And there will be certainly lots of scope for undergrad projects to explore that using these kind of techniques, maybe not with a very dangerous laser setup, because it needs a lot of very specific training, because like I can't turn that on without putting the magnetic lock on the door. That's why I didn't show it running, because I'd have to be wearing crazy goggles and all of this. But using certainly fluorescence spectrometry, the thing I actually just demonstrated there, like that, these are people use these techniques in their PhD, their master's projects, and there's no reason why that couldn't be in an undergrad project as well. If you were interested in protein therapeutic formulation, I guess is the long answer to that one.
That's great. And I just think it's worth pointing out one of the things that undergraduates do say to me is that they get a chance to actually use our labs if it's something they're interested in. There are also summer research projects which they can get involved in. There's the iGEM as well. You can find out more about that by watching Steffi's film. But I think
another thing I suppose I could mention is that, like, all of the control software for the instrument that we built in there was like, we had to develop all of that. Like it was developed in-house, and there is scope for, if people are more on a coding side and you're interested in developing software for, like, you know, real instrumentation control, process control, like there's kind of scope for that as well. Like, there's a question.
Cool. Okay, that's that. Do you want to read it out, or can you get that on your screen?
I have to click a button, but I can do it.
Well, I'll read it out to you. So, what will be the difference shown in decay rate between a denatured and a normal protein?
Right. So this is a very interesting question. It doesn't have a very easy answer though, because it isn't. I think what they're asking is, would the decay be longer or shorter? And I think that would be probably the most sort of like how do I say it, the not the simplest way, the most intuitive way of looking at it, that would increase or decrease, but it isn't actually that. It's just that it would change. Like, you can't really tell, because if I bring up my diagram from before, like the difference in how long this takes is how, like, to go from this excited state to a slightly less excited state so it can emit the photon is dependent on vibrational changes. And like, you can see one of the things about protein structure is that it's very complicated. Like, as it denatures, it will, you know, the thing will tend to be pulled apart. Will that mean that there's something vibrating close to this or further away? And you can't really tell that. It's not as simple as it would increase or decrease. It was just that you would be able to make a relative measurement from the beginning to the end. Like, if I was denaturing a protein by exposing it to temperature and I was continually measuring the fluorescence lifetime, the decay rate would change. Do you know what I mean? Like, but it would always have to be then a relative measurement or a reference measurement. You'd have to know the prior decay rate. It's not that you could just predict, oh, it's going to move in this direction or that direction, which it's kind of, maybe that isn't very intuitive, but it is because protein structure is such a complex thing that we can't even, well, you know, AI is starting to be able to sort of like predict it, but not perfectly. Do you know what I mean? And like, solving protein structures is one of the big challenges that kind of remains in science. Like, it's a good question.
Great, thank you very much for that. Well, that's the only question we've had, and we've used up our time. So I'm just going to say that if there are no other questions, I'm going to wrap this up and say thank you very much for joining us. It's been great to have you. Huge thanks to Sam for showing us around the lab. And
yeah, no worries.
And
if anyone's got any further questions later, get in touch with Kim. And if everyone's got any questions they think of later, get in touch with Kim, throw them at me, and yeah, we can chat about it whenever, you know.
Thank you very much. Really appreciated. Great. Thank you very much. Thanks for your kind words in the chat. But have a great rest of the day. Have a fantastic holiday. Have a really good break. And yeah, all the best. Thanks, Sam.
Yeah.